Hello, I'm Tobias Jores, and I'd like to tell you about my work on learning the grammar of plant regulatory DNA. And I want to start this with a look in the future. And so predictions by the United Nations say that by the end of the century, there will be about an additional 3 billion people living on Earth. However, we're living also in a phase of climate change. And so if we look for the same time span at predictions of crop yields, we can see that the yields of most of our main crops are actually predicted to drop in response to climate change. And so to ensure food security, we actually need fast and efficient methods to generate crops that are both stress resistant and high yielding, even in the face of climate change. And so luckily, I think that the technologies to do this are starting to emerge. And I think especially genome engineering will be a key player in this effort. And so the capabilities of genome engineering have recently been demonstrated by the rapid de novo domestication of several different plant species. And so while this technology is really coming to maturation, what we also need is good targets that we can engineer in order to improve crops. And here I want to make a case that cis regulatory elements might be very promising targets for genome engineering. And in fact, a review that was published in 2013 found that about half of all the domestication alleles that we know reside in regulatory regions. And so in contrast to mutations in the coding sequence, those in cis-regulatory regions can lead to tissue or condition-specific effects, and they often show a graduated response. However, they can also be very powerful. And so to illustrate this, here is a picture of teosinte, one of maize. And we know that during the evolution, a transposable element jumped in front of the TB1 gene in the maize genome. And because of this transposable element, the TB1 gene is now expressed at higher levels. And this leads to many of the striking morphological differences between the two plants, and which in the end really is the reason why maize is such an efficient crop for us nowadays. The problem, however, with targeting cis-regulatory elements for genome engineering is that they are very poorly annotated, and the regulatory code that governs their activity is largely unknown. And so I've made it my mission to try and make plant gene regulation predictable, because I think that really this good and deep knowledge of plant gene regulation will enable rapid crop improvement by genome engineering. And so to do this, I've developed an assay that we term plant STARR-seq. And so here we're using a GFP reporter gene that has a DNA barcode at its 5' end. This reporter gene is driven by a 35S minimal promoter. And this reporter construct can further be modified by adding cis-regulatory elements upstream of the minimal promoter. These elements, for example, enhancers, can then be linked to the barcode by next generation sequencing. And all those different constructs can be pooled and used for transient transformation of tobacco leaves in a library fashion. And after 2 days in the leaves, we can then extract the mRNAs of the reporter gene and sequence the barcode both in the RNA as well as in the DNA input. And this will give us a table how often each barcode was detected by next generation sequencing, either in the DNA input or in the RNA output. And we can then calculate the enrichment of a barcode in the RNA. And this enrichment is a direct measure for the strength of the associated enhancer. And so to show this it actually works, in a pilot experiment, I tested the activity of the viral 35S enhancer as well as of 3 native plant enhancers. And for all 4 enhancers, we could clearly distinguish their activity from that of a control construct that doesn't have any enhancer. And so after showing that the assay works, I decided to look at cis-regulatory elements genome-wide. And so I started out with plant core promoters. So for the study, we defined the plant core promoter as a region that starts 165 base pairs upstream of the transcription start site and extends to 5 base pairs downstream of it. And so we took these 170 base pair sequences of every gene in the genome of Arabidopsis, of maize, and of sorghum, and we subjected them to arrays of this. And so the synthesized core promoters were then cloned into our STARR-seq reporter construct and replaced the 35S minimal promoter. So now it's this core promoter library that is driving the expression of the reporter gene. And I was also curious if these co-promoters can interact with additional cis-regulatory elements. And so I created a second library where I cloned the 35S enhancer upstream of the co-promoter library. These libraries were then again tested in a pooled fashion by transiently transforming tobacco leaves as well as maize protoplasts. And so here I want to give a shout out to my colleague Jackson, who did all the protoplast work. And irrespective of the assay system, in the end, we can extract the mRNAs from the tobacco leaves or the maize protoplast and sequence the barcode in both the RNA as well as in the DNA, calculate the enrichment of the barcode in RNA, and this will be a direct measure for the strength of the associated co-promoter. And so here is a summary of the results that we got with the results from the tobacco leaf system on the left and the results from the maize protoplast on the right side. And at the bottom of the plot, I'm indicating the number of co-promoters that are in all of those groups. And so if you sum them up, you can see that we were able to measure the activity of more than 75,000 co-promoters. I also want to point out that the promoter strength on the y-axis is on a logarithmic scale. And so in all cases, we have a wide dynamic range of promoter strength, with the strongest promoter being more than 250 times stronger than the weakest ones. And so on the slide before this, I just showed you that we also tested these co-promoters in conjunction with the 35S enhancer. And we can see that the activity of almost every single co-promoter is stimulated by the addition of the 35S enhancer. So we can even measure interactions between individual cis-regulatory elements like promoters and enhancers. And so now we have this wealth of data, and to make efficient use of it, we turn to computational modeling. And so this was done together with the lab of Ed Butler and his PhD student, Travis Reitzman. And so we trained two convolutional neural network models on the data from the tobacco system, as well as on the data from the maize system. And we then used those trained models to predict the activity of a held-out test set. And so this test could show that the models can accurately predict promoter strength with an R-square of up to 0.7. And so to highlight the usefulness of such models, we decided to use them for a process that we term in silico evolution. And so for this, we can take any promoter sequence and then digitally create every possible single base substitution variance. All these variants can be scored with the model, and we can look for the variant that has the highest predicted strength. And this sequence is then subjected to another round of this process. So in each round, we will introduce a single mutation in the promoter, and with each round, this should make the promoter stronger. And so to test if this actually holds up in an actual experiment, we chose about 200 promoters that span a wide range of activity in both our model systems. And then we subjected them to 3 rounds of in silico evolution and retested their strength. And we could see that these 3 mutations that we inserted clearly had a beneficial effect on promoter strength. And we could also see that if we did 10 instead of 3 rounds of in silico evolution, we could further increase promoter strength. And so to really drive home how powerful this approach is, I want to point out that in our starting set, we only had 6 promoters that were stronger than the viral 35S minimal promoter. However, after the in silico evolution, we had now more than 100 promoters that were stronger than this highly evolved viral promoter. And so after this study that is actually published already in Nature Plants on co-promoters, I now turn to measuring the activity of enhancers. And so to do this essentially in a genome-wide fashion, we used a little trick. And so we know that enhancers work by recruiting transcription factors, which can then interact with the core transcription machinery. But in order to be able to do so, those enhancers have to reside in accessible chromatin regions. And so what we can do is we can take accessibility data, and from this look for regions that are accessible and might indicate that they have enhancer function. And so what we did in the end was we took the accessibility data for 4 different species, Rhodopsis, tomato, maize, and sorghum. And then from this, we selected over 175,000 accessible chromatin regions that might have enhancer activity and subject them to array synthesis. And so these enhancer candidates were then again cloned into our reporter construct upstream of the 35S minimal promoter. And we actually cloned them in both the forward and the reverse orientation, essentially doubling the number of test sequences. And all those sequences were then again pooled and transiently transformed into tobacco leaves. And so for all of the experiments, we actually did 2 biological replicates. And you can see that if we look at the replicate correlation, this is really high with an R-square of 0.86 or 87 in both tobacco leaves that were kept in the dark or in the light, showing that we can really reproducibly measure the strength of enhancers even in such a big library. And then if we look at the activity of the different enhancer candidates grouped by species, we can see that clearly the enhancer sequences from the dicots Arabidopsis and tomato perform better in tobacco, which is itself a dicot, than the ones from the monocots maize and sorghum. We can also see that across all cases, we again see a wide dynamic range of activity. And as I said before, we have measurements for over 350,000 elements. So again, a very big data set. And so again, we also thought computational modeling might be very interesting for this data. And so in a very simple experiment, I tried to see what happens if I use the CNN model that was trained on the core promoters to essentially predict promoter strength, and to see if this is correlated with the enhancer strength that we measured now in the assay. And so you can see with an R-square of 0.15 or 0.17, the accuracy is not very high. And I think this just means that while promoters and enhancers do share some elements, I mean, there is some correlation, there are also elements that work specifically when in the context of a promoter, but that are inactive when they are in the context of an enhancer. And so really showing that promoters and enhancers are distinct elements, at least to some degree. And so next, I used a model with the same architecture, but this time I trained it on the enhancer data and then used it to predict the activity of a healthy test set. And so now you can see that this model has a much better accuracy with an R-square of 0.55 to 0.6. But you can also see that the model is still under-predicting the activity, especially of the strong enhancers. And so future work on this model by maybe changing the architecture will hopefully help to further increase the accuracy of that model, especially for the strong enhancers. And so I showed you now that we can, for more than 75,000 genes, assign promoter strength to all of those promoters, and we can actually predict the strength of a promoter of any arbitrary gene that we haven't measured yet. And so since my newest experiments, we can actually add a second column to this table where you can now assign also the strength to enhancer elements. However, here the problem is that an individual gene will always have just one co-promoters, but it might be linked to several different enhancers. And so this will make the follow-up work harder. And so for now, I've decided to just use only the strongest enhancer and ignore the other enhancers that might be linked to a gene. And so with this STARR-seq data, we can now ask the question if it is correlated to plant gene regulation. And so we can take RNA data seeds from different parts of flowers, from different parts of plants like Arabidopsis and maize, and see if this expression, the expression level of the different genes as measured by RNA-seq is correlated to our STARR-seq measurements. And so when we do this by using our promoter data, we can see that the predictability for gene expression is not very high. And it's actually interestingly, it works better for maize than it does for Arabidopsis, even though evolutionary, the tobacco system which this data was generated is closer to Arabidopsis than it is to maize. Interestingly, when we instead use our enhancer data to predict gene expression, we can see that the correlation is higher this time for Arabidopsis, which makes sense because those are the two that are more close in evolutionary time. But we can also try to see what happens if we take both the promoters and enhancers. And so what I chose to do, again, a very simple model, is to just add up the two values we got from STARR-seq and see how well this correlates with gene expression. And here we get on average the highest correlation. And we can see that they're actually relatively similar for both Arabidopsis and maize. And when we look at this a bit more close by separating the RNA-seq samples and whether or not they were generated in a leaf sample, we can see that in Arabidopsis, the highest or the RNA-seq data that is best correlated to our STARR-seq data is the one that's coming from leaf, which makes sense because our STARR-seq data was also generated in leaf. So it might even capture some of the tissue specificity that's kind of inherent in the system. However, overall, if you look at this, you can see that the highest correlation we achieved so far is in Pearson's R of 0.25. And this is clearly not good enough to really use this prediction, to use it as a predictive model. And so I think we need future work to make this more predictable. And so I think there actually is a good chance that this is doable, because so far I was really using a very simple model where I'd used just the activity of one single enhancer in a core promoter and used a simple addition to kind of combine their strength. And when in fact we actually know that there's way more elements controlling the expression of the gene. Not only are there more enhancers, there might also be insulators or silencers that can also affect the gene expression. And so we are currently working on identifying and characterizing such elements. And so together with Cole, who is a postdoc in our lab, I'm working on using STARR-seq to measure the activity of both insulators and of silencers. And in parallel, Sai, who's a PhD student in the lab, is working on measuring the activity of cleavage and polyadenylation sites on transcript levels. And so the more elements we study, the more comprehensive our understanding of gene regulation will be. And so additionally, several people in the lab are actually working to generate accessibility maps using techniques such as single-cell attack or fiber-seq to get both cell type specific accessibility, but also co-accessibility of cis-regulatory elements. And I think this data will be really useful to understand which cis-regulatory elements are active in which tissue, to then also enable us to predict gene expression in a tissue-specific way. And of course, once we have all this data, we kind of have to pool all of this together and use some machine learning model that can then accurately predict gene expression. And so here I have to admit that myself, I'm mostly a wet lab person. And so I really do not know which machine learning model would be best suited for this task. And so if you have ideas for this, please feel free to reach out if you want to collaborate with us on this. If you want to take a look at the data, we are very happy to work with you. And so to sum up what I just showed you is that I've established this plant STARR-seq assay that can identify cis-regulatory elements. And it actually works across many different cis-regulatory elements. And so we are now seeing is as the one assay to find and also to characterize all cis-regulatory elements in a plant genome. We could show that this assay is sensitive enough to detect both condition and species-specific activity, and it generates a wealth of data that's ideally suited to both predict but also evolve the activity of those elements, at least individually. But I think the real goal in the future, and I think there's potential for this, is that we can use this STARR-seq data to actually predict in planta gene expression. But again, I want to point out that this is still in infancy and that we would like your input on this. And so if you want to collaborate us both on the computational modeling side, or if you want to do STARR-seq for your favorite gene or in your favorite organism, please feel free to reach out to us. And so I quickly want to thank both the members of the fields and the Quite Lab, our collaborators and my funding sources. And I want to give a special shout out to all the people that are working together with me on Plant STARR-seq. Thank you.